Bom dia, bem-vindos todos a essa reunião geral do Departamento de Psiquiatria do Hospital das Clínicas da Universidade de São Paulo. Hoje a gente vai ter uma reunião muito especial, com um convidado muito especial também, o Dr. Gregory Fricchione. Ele é diretor do Benson-Henry Institute for Mind-Body Medicine e do Chester Pierce Division of Global Psychiatry e co-diretor do McCance Center for Brain Health no Mass General Hospital. Ele se graduou pela NYU e é professor de psiquiatria da Faculdade de Medicina de Harvard. Ele vai falar um pouquinho hoje para a gente sobre Mind-Body Medicine and the clinical implications of brain evolution. So, welcome, Professor. I was just welcoming everybody, our audience on YouTube. We are honored to have you here with us today to be able to learn from you. So, let's start it, and then we encourage our audience to, if they have any doubts or something, to make them on our YouTube chat so we can give it to you later.
Okay. Well, thank you, Dr. Mazzocco, and I want to extend my thanks to my dear friend, Professor Manuel, and to your new chairman, Professor Forlenza, for inviting me here today to speak to you. I'm going to share my slides now. And hopefully you can see. How's that? Can you see?
We can see it, Professor, but I think that if you put like presentation, we will be able to like this.
Is that right? Okay, good.
Perfect.
All right, here we go. Just the usual kind of honoraria and travel and some royalties. So we have a lot of things to talk about. We're going to explore our major health challenges around the world, Brazil and the United States included. And we're going to talk about stress, and we want to talk about brain evolution and how it is a wonderful way for us to understand neuropsychiatry. We're going to talk about the damaging effects of chronic stress for your health. We're going to introduce the concept of resilience. I'm going to offer up a hypothesis that there is a stress to resilience ratio that we can use in clinical medicine. I'll talk a little bit about candidate research areas, very exciting to those of us interested in the relationship of mind, brain, and body. And then we'll offer up a synthesis of about what this might all mean, and why it's important to link up our role as clinicians with an increasing role we have in public health. And by that I mean health promotion and illness prevention. And certainly psychiatry, because we're doctors of the master gland, aren't we? This is extraordinarily important. We have a large role to play in trying to promote the health of our communities and prevent illness, and not only psychiatric illness, medical illness as well. Let's see. So what is mind-body medicine? It's that part of medicine which recognizes that brain and body are one, and it uses evidence-based effects of thoughts, beliefs, and emotions to positively influence physical health. And in order to promote health and healing, it employs a heterogeneous, a varied group of techniques, including relaxation, breathing relaxation exercises, progressive muscle relaxation included, meditation, biofeedback, guided imagery, hypnosis, yoga, tai chi, qigong, autogenic training. And I think mind-body medicine works off of three major hypotheses. The first is that the environment is biology and mind-body is a unity. You really can't separate out what your experience is as you hear my voice and biological events in your brain. And then this leads to the second hypothesis, that it's psychosocial stress creates vulnerability when it leads to cellular oxidative stress and loss of mitochondrial reserve capacity. And we'll get into that today. And then that's that cellular oxidative stress with what's now being called allostatic load, and we'll talk about allostasis and allostatic load in a moment, that's what leads to disease vulnerability. And again, I stress the point that we're talking about psychiatric illness as well as medical illness. So this is an equal opportunity process where overwhelming or toxic stress, if you have low resilience, will cause disease vulnerability. That's the core of mind-body medicine. So what are our major health challenges all around the world? Well, most people would agree that stress-related non-communicable diseases are the major challenge. And of course, we are in the midst of a horrible pandemic that has stricken our two countries to perhaps an oversized degree, unfortunately. But still in all, stress-related non-communicable diseases before the pandemic was killing over 60% of the world's populations. And we're talking about cardiovascular diseases, chronic pulmonary and GI diseases, diabetes, arthritis, and neuropsychiatric disorders. So there's enormous morbidity and mortality. And of course, it also contributes to the ballooning of healthcare costs. So many people believe that this is the global health challenge of our century. And this slide, which comes from the Disability Adjusted Life Years literature, shows you that neuropsychiatric disorders are close to 30% of these chronic non-communicable diseases. And of course, neuropsychiatric disorders are often comorbid with many of these other non-communicable diseases. So our role as psychiatrists and neuropsychiatrists is extraordinarily important in battling this major health challenge in the 21st century. Now, one of the problems we face is that there's what some people call the inverse care law. And this is, I'm using the phrase of Julian Tudor Hart, publishing in The Lancet. He wrote, "The availability of good medical care tends to vary inversely with the need of the population served." And so the point here is that one driver of the NCD epidemic is socioeconomic status, and this comes from the pathological effects of poverty, but also the lack of access to medical care, medical and psychiatric care. You'll see in a moment that we have a hypothesis that the same core risk from physiological disturbance it increases vulnerability not only to stress-related non-communicable diseases, but to viral illnesses. And this is the work of Sheldon Cohen several years ago. And we know this in the United States. We have these things called zip codes, and we can tell an awful lot about a person's risk profile for these stress-related non-communicable diseases simply by knowing what zip code they live in, what community they live in. And this is a real example of the disparities that we face in the United States in terms of healthcare and life expectancy. So do you know that in Boston, the African American community predominantly lives in Roxbury, and in Roxbury, the life expectancy is 58.9 years, which is shorter than that of many third world countries. And it's similar to the life expectancy of an average American back in the 1920s. So this is very powerful, and it's also very provocative about what we need to do in our country to reduce this terrible disparity in healthcare. And we're seeing the same disparity with COVID-19 in our country. And so what is the common linkage between chronic stress-related non-communicable diseases in terms of risk and COVID-19 in terms of risk. We've just published a paper in brain behavior and immunity on this hypothesis. And when you think about immunology, there are three terms that are coming into vogue. One is damage-associated molecular patterns. So when there's any kind of damage to your cells, there's structural breakdown, and that leads to the presentation of antigens, self-antigens, but changed self-antigens, okay, but also foreign antigens. And when it's a foreign antigen, it's called pathogen-associated molecular patterns, PAMPs. When it is a self-damaged antigen, it's called non-pathogen-associated molecular pattern, non-PAMPs. And what we're discovering, and this is so powerful for all of psychiatry, is that psychosocial stress, if severe enough, is going to lead to the production of non-PAMPs, non-pathogen-associated molecular patterns, that become prompts, become triggers for an innate immune response. And this innate immune response can be taking place in the periphery, but also in the CNS. And when it occurs in the CNS, people are starting to call that neurogenic neuroinflammation, which we'll talk about. But as you can quickly see, that we have to readdress all of our psychiatric pathologies with this new information. Because when we're talking about schizophrenia or bipolar illness or major depression or anxiety disorders like post-traumatic stress disorder, we have to account for the fact that it's very likely that psychosocial stress has led to neurogenic neuroinflammation. And so our simplistic hypotheses about disordered synaptic functioning and neurotransmitter changes, etc. Yes, there's validity in using that strategy to understand neuropsychiatric illness. But now we have to account for the very powerful effects of microglial inflammation in the brain when there is intense psychosocial stress. Now, with COVID-19, there's something very interesting to pay attention to, and that's something we call the conserved transcriptional response to adversity, CTRA. And UCLA is a hotbed of research into conserved transcriptional response to adversity. What do I mean by that? So when you have a chronic stress situation, perhaps the driver for the development of chronic non-communicable diseases, heart disease is a perfect example, that is at its core a reflection of a change in your transcriptome. So there it is, as you know, we know now that while you're born with a certain genetic endowment, your environmental experience affects the activation or relative deactivation of your genes. And so this transcriptome effect is very powerful because it leads to a change in the proportion of proteins that your cells are producing, right? So we can look at the messenger RNA and understand transcriptome changes related to the environment. And if you're living in an environment with severe psychosocial stress, then you're going to affect a change in your gene expression. And that's at the core of this idea of the conserved transcriptional response to adversity, to stress. And what happens is that the transcription factor NF-kappa B gets upregulated under conditions of psychosocial stress, and that leads to the production of a heightened level of pro-inflammatory cytokines, IL-1, IL-6, TNF, a variety of powerful pro-inflammatory cytokines start circulating in your system, not because you're facing an infection, because you are facing a psychosocial stressor that leads to a change in your gene expression through an upregulation of the transcription factor NF-kappa B. But another thing happens when you're facing chronic stress, and that is a reduction in the antiviral transcription factor interferon type 1. And so when we have in the United States the very clear finding that our Black population, our Hispanic population, our indigenous population, all have 2 to 3 times the risk for severe COVID and death, we have a hypothesis to consider, and we hope to research this with the UCLA group in the near future, because it may be that in those minority populations, because of discrimination stress and disparities in experience, that minority populations wind up having an elevation of their baseline innate immunity and a reduction in their baseline antiviral immunity. That elevation in innate immunity leads over time to the development of cardiovascular disease, chronic pulmonary, chronic GI disorders, neuropsychiatric disorders, etc. And that's what you read in the literature right now. People say it's the social determinants of illness that is causing the increased risk to these minority populations in the United States. But really, we believe, our hypothesis is that it's that same core conserved transcriptional response to adversity and stress that's leading both to the non-communicable disease risk and the elevated risk to severe COVID-19. So I spent a lot of time on that, but I think it's very topical. And I would be interested to hear whether you're seeing similar challenges in Brazil. And this is the paper that just came out in Brain Behavior and Immunity, putting forth this hypothesis. I'm going to skip through some of these slides that I just talked about. Now, what about these non-communicable diseases in relation to stress? I put up this slide. This is a famous study in the United States. It's the Life Stress Unit Scale study of 2 psychologists, Thomas Holmes and Richard Ray. And a while back, they were able to go to the Pacific Northwest of the United States and talk to people without psychiatric history about what really caused them the most stress. And they asked them to put a numerical value on different experiences. And I show this slide because, as you see, 8 or 9 of the top 11 stressors are separation events. And this becomes very important for our understanding of what stress really is. And so here's my definition of stress. Stress is what the brain does to itself and the rest of the body when faced with a separation threat or challenge, endangering one's attachment. And so you'll understand when I give you that definition that I'm going to spend time focused on attachment theory, classic Bowlby in terms of the relationship in human beings of how challenging it is to face separation and how we crave secure attachment. Now, there are three different types of stress. There's normal stress. So when you wake up in the morning, you have a bump in your cortisol because your body understands that you need that stress hormone because life, the experience of living, requires some stress. And that's normal. You're thankful that you have some stress because it means you're alive. And if you come to work one morning and say you're working in the emergency room at USP and you come to work and there are 40 patients in the emergency room like there was at Mass General Hospital on Monday, you're going to move from normal stress to tolerable stress, hopefully tolerable and not toxic. But if there are 40 patients who are all terrible cases, you may overflow into toxic stress land. And if that was to continue for days on end, like it has for some frontline clinicians dealing with COVID-19, you may wind up in a chronic state of toxic stress. So if you look over at this bell-shaped curve, this inverted U curve, a normal distribution, if you will, a Gaussian normal distribution curve, in this green area on the left side, the upslope, we're referring to normal stress. This is what I mentioned. If you wake up in the morning and you're facing a normal day, you go from being inactive to being somewhat active and it feels fine. And then your boss tells you you have to get your project done in 10 hours. You may go into tolerable stress where it's going to increase. But then if you go over the top into the toxic stress range, you may wind up exhausted and develop anxiety and break down into depression. And so on the x-axis, you have your stress level. On the y-axis, you have performance or your quality of health level. This is really important as a simplistic way of understanding our stress curve. And when I mentioned the conserved transcriptional response to adversity, to the transcriptional response to stress, you can understand that if you're in the red zone, you're going to have a gene expression profile very different than if you're in the green zone. And that really is what leads to your illness vulnerability. So what is allostasis? Allostasis actually comes from physiologist Sterling and Eyre writing in 1988. And they were trying to get across the understanding that our bodies, our brain and our body seeks to keep our physiological systems, our hormonal axes as well, within a normative range. But we live in a changing environment, so we want to maintain that stability in the face of continual change. That means we have to have some fluctuation, but we don't want to spend too much time, a standard deviation or two outside the norm. We want to keep it in a normative range as best we can. So we don't want a system where our blood pressure is always 120/80 because sometimes we need more blood pressure. We don't want to have a heart rate that's always at 65 because sometimes we need more heart rate. We don't want a thyroid axis that just puts out a certain amount of thyroid hormone because sometimes we need more, sometimes we need less. That's the concept of allostasis. It's your brain that's doing that work. And occasionally, if you are in the toxic stress range, more often than not, your brain works overtime to manage your physiology. And in the process, it expends an awful lot of energy and creates an awful lot of oxidative stress. and your mitochondria develop stress and start to perform inefficiently. Therefore, you wind up with disease vulnerability. I want to also introduce, and that's called allostatic loading. And at the mitochondrial level, that's called mito-allostatic loading. This term hormesis, you're going to hear more of. And it simply is a term to describe a bell-shaped curve, biphasic. And stress has a biphasic effect on your whole brain and body. And so life without stress is not a good thing, because you need to get used to stress and stressful events. And the term stress inoculation is used for that. And post-traumatic growth is a part of that as well. But if it's chronic, if you've got chronic high-level psychosocial stress, that can lead to disease vulnerability. Here's the brain that's doing this for you. And I want to point out this pink area, the amygdala. You're all familiar with the amygdala, down in the medial temporal lobe region. Very ancient structure, vertebral structure, that's necessary for life, for self-preservation, because it is your fear conditioning center, and it was built by evolution to create a threat signal. And when you're uncertain about something, it could be a threat to you and your survival and your preservation. So your amygdala pours out glutamate and excites the rest of your brain to be vigilant, because it may indeed be a threat. This is an important point, because the way you're built, you are going to have a hair trigger system that is amygdala driven. And it works in two ways. It will set in motion an acute stress response. And so you're going to have cortisol pouring out of the HPA axis. The adrenal cortex will put out cortisol. You're going to have the sympathetic nervous system outperforming the parasympathetic nervous system, and a lot of epinephrine from the adrenal medulla and norepinephrine from the locus ceruleus pouring into your body, making you more activated and vigilant. But you're also going to have an innate immune response. And that acute inflammatory response coincides with the acute stress response. And that's what is depicted here in this slide. So your amygdala gets threatened. There's fear, anxiety, worry, hypervigilance, avoidance, and importantly, emotion dysregulation. Emotion dysregulation is a part of so many of our psychiatric disorders, isn't it? But fortunately, you have evolved this prefrontal cortex, importantly, the dorsal anterior cingulate, we're now calling it the mid-cingulate, and the ventral anterior cingulate, which is part of the medial prefrontal cortex, along with the orbitofrontal cortex and the ventral medial prefrontal cortex. These areas of the brain are effective in normal brain in talking to the amygdala through GABA interneuron blueprinting to reduce the output of the central amygdala and to reduce that excitatory to inhibitory imbalance for the rest of the brain. That promotes health, and not only brain health, but also the health of the whole body. Because if you're living life with an excess of cortisol, or an excess of sympathetic drive, or an excess of innate immune response, you're, as we say in the United States, you're cruising for a bruising. You are creating excess vulnerability to all forms of illness. And we know that there are long-range effects of being in that state. So because that prefrontal cortical gray matter volume is at risk if you stay in a state of chronic toxic stress. When there's greater subjective chronic stress and cumulative adverse life events, there's smaller volume in the medial prefrontal cortex and anterior cingulate, and in the anterior insular cortex, which is also part of the paralimbic network of prefrontal cortical structures. So this is essential to emotion regulation. And these wonderful slides, one from Ross in JAMA Psychiatry and the other from Jovanovic and Carrie Ressler at Mclean. And you see, again, this very important part of the medial prefrontal cortex and being able to dampen amygdala tone. The hippocampus, by the way, an ancient structure along with the amygdala, can also, to a lesser degree, inhibit amygdala tone. And this is from the Jovanovic and Ressler article. And you see the blueprinting here with the intercalated GABA interneurons that can turn off the central amygdala outflow. So I'm going to skip this, but the point is that the evolutionarily based inflammatory bias and the evolutionary based social threat bias can interrelate. And so you can have an enormous number of false positives that the amygdala drives that lead to a kind of vicious cycle where your innate immune response and your acute stress response, even if the threat is not real, can lead to disease vulnerability. So where does this come from? I want to give a shout out to Nikos Tinbergen, a real hero of mine. And Tinbergen, when I think about him, he made such an important contribution. He won the Nobel Prize in 1973, but in a wonderful article in 1963, he was an ethologist who, along with Konrad Lorenz, they were the two major ethologists of the 20th century. And he basically said, well, if you want to understand anything biological or psychological, you have to ask yourself 4 questions. How does it work? How did it develop? What is it for? And how did it evolve? Okay? And when I teach my residents and fellows, I'm always bringing that up. I'm always pointing them towards those 4 questions. This is an antidote to the nosology we use in DSM. And what do I mean by that? What I mean is, in my experience, and I'm a neuropsychiatrist working in consultation psychiatry, the patients that I see are most of the time much too complex for DSM. Otherwise, you're kind of layering all of these different so-called diagnoses on top of each other because they're so complex in terms of what's going wrong with their brains and their body. Tinbergen is a refuge for us working in that environment because we can bypass all of that 16th century nosology because that started with Sydenham back in the 1600s. It's very ancient to cluster symptoms and think that you're actually looking at something truly biological. But if you use Tinbergen, you can return to first principles in understanding what's going wrong with that patient. So that I think is a way to start the evolutionary discussion. Now, very quickly, every living entity shares 4 operations. It senses its environment, it analyzes the incoming data, and it affects a motor response. Any living thing you tell me about, I can analyze it with those 4 operations. So Kieran Smith, biologist from the UK, a while ago said that every living organism is a sensory motor analyzer effector. So the brain evolved as an organ designed by natural selection to specialize in the organism's sensory analyzer effector and motor functions. It's very important to understand this. So when you're evaluating every one of your patients, you're a brain doctor, and you go into the situation understanding what the brain evolved to do. The brain evolved through natural selection to sense your environment, to analyze what you sense and perceive, and then to decide on a motor response. Just to show you, this is a unicellular organism, E. coli, and Karen Smith depicts it in this manner. There are receptors on the surface of E. coli, which tells it, okay, there's a concentration gradient here of amino acids or sugars or dipeptides. I like that stuff. It analyzes that concentration gradient, and then it braids up its flagella and it moves toward it in an example of positive chemotaxis. Now, if I substituted here TNF alpha or an interleukin, what do you think would happen? E. coli would analyze that, and it would unbraid, unfurl its flagella, and it would try to tumble away. So in a sense, E. coli has primordial intelligence. It is sensing its environment, it's analyzing the information, and it's affecting a motor response. And bacteria understand the need for attachment. This is very powerful, and it's a process called quorum sensing. So single-celled bacteria in a certain environment, in a context, will communicate with one another, and they'll form a biofilm if it serves their purposes. So there's a hint there about the meaning of evolution for biological organisms, and they use many of the same molecules. So intercellular adhesive molecule is used by bacteria to form attachment solutions to their separation challenges. This is a green turtle brain, a reptile brain. And very simply, there's a hippocampus that's dorsal, and there's a general pallium, and there's a primitive piriform cortex and a big, gigantic olfactory bulb. That's a reptile brain. But if I was to actually see all of you, I would ask you this simple question: are you a mammal? And you would all kind of smile and maybe gingerly raise your hand. Yes, you're a mammal. These are two beautiful mammals that I show here. And when I know that, when I know you're a mammal, I know what your survival strategy is. To a large extent, your survival strategy, which started about 300 million years ago, your survival strategy really was to attach. Parental nurturance and social attachment became your survival strategy. And part of the reason for the reason why you have that capacity is because form follows function in this case. And here you have this stippled area. This is an early mammal brain. This is your cingulate cortex. And only mammals have cingulate cortex. Very mammalian. And then we have to introduce this advance by Garrett Alexander and in the neurology department at Hopkins in the mid-'80s, that really the brain is made up of these parallel segregated circuits that do differentiated function to a very high degree. And there's meaning embedded in the structure itself, because these are basal ganglia thalamocortical loops that do certain functions in our brain. But in that terminology, you know that this really just reflects the fact that the brain is a sensory motor analyzer effector. Because when we talk about the thalamus, it's your sensory part of the brain. When we talk about the basal ganglia, it's the motor part of your brain, simplistically speaking. When we talk about the cortex, it's your analyzer effector part of the brain. And in order to, the part that makes you really human, a mammal with higher consciousness is this anterior cingulate basal ganglia thalamocortical loop, because that helps you valence your experience. It gives salience to what you're experiencing, and that will play a large role in your decision to affect a motor response. And your anterior cingulate actually becomes a hub of decision making, especially when you're conflicted. Maybe your emotions and cognition are not in concordance. And I like to show this slide. Here's your anterior, this is your mid-cingulate. This is your, used to be called dorsal anterior cingulate, your ventral anterior cingulate. And here's your posterior cingulate. And so this area, along with the orbitofrontal cortex and ventromedial prefrontal cortex, is going to speak to the amygdala through those tracks I showed you and dampen amygdalar tone. And this, of course, is part of what mind-body medicine is doing. It is helping this part of your brain do emotion regulation. And through these evidence-based approaches, meditation, mindfulness, but also cognitive behavioral therapy, positive psychology, optimism, purpose, meaning, this part of your brain gets stronger. Acceptance and commitment therapy, this part of your brain gets stronger and has a better capacity to dampen your amygdalar tone. Because if the amygdala gets upset, it's going to speak to your hypothalamus. It connects up with your hypothalamus, connects up to the paraventricular nucleus, which sets in motion the HPA axis. It connects up with the lateral hypothalamus, which connects up with the locus ceruleus and puts out a hypersympathetic tone. And it also connects up with another part of the hypothalamus, mediobasal, which helps with the development of innate immunity. Now, this is another hero of mine, Paul Mclean. Paul Mclean was one of the 20th century's most important comparative neuroanatomists, and I had the privilege of spending time with him back in 1990 when I had a sabbatical. And Paul knew everything there was to know about lizard brains, bird brains, fish brains, mammal brains, primate brains, and human brains. And he basically said, the most painful mammalian condition is separation, exclamation point. He was the father of the limbic system. He's the person who put the limbic system on the map. And here's Bowlby. Bowlby said man's environment of evolutionary adaptiveness is always one of secure base attachment. And I believe he was correct. And so your mammalian anterior cingulate is the key to the attachment solution. And I'll show you in a moment the anatomical reason for this. But Paul Mclean talked about the mammalian behavioral triad. Mammals do maternal nurturance well. The pups of every mammalian species have a separation cry when they're separated from their caregiver, and social play. All of that is being mediated in the anterior cingulate. And there are 4 attachments that are important for the human being. 2 of them are shared with vertebrates, and that's attachment to food as a source of metabolic energy, attachments to sexual objects, so you have self and species preservation. And then when you become a mammal, you have attachment to parental and social objects. And then when you become a human being, you have this great expanse, the frontal polar cortex, dorsolateral prefrontal cortex. These areas of your brain connect up with your memory system, your memory of the past system, and your hippocampus and so on, to develop this magical capacity we have to have a memory of the future, to do future planning. So all of you in the audience there in Sao Paulo, I know that you have an amazing frontal lobe system that allowed you to plan, to have a memory of your future, or else you wouldn't have had the ability to stick with all of the training and schooling that you needed to get to where you are. as a physician and as a psychiatrist. Whenever you're separated from any of these four core attachments as a human being, you suffer. And that becomes the kind of engine for neuropsychiatric disorders. It's the engine for all stress-related non-communicable diseases. And your brain craves an attachment solution because of what Bowlby taught us. Now, this is from Marcel Messelam, the great behavioral neurologist now at Northwestern and emeritus. Vertebrates have two ancient moieties or pathways. There's a dorsal one, it's the hippocampocentric. There's a ventral one, it's the olfactocentric. The dorsal one tells you where you are in space. The ventral one tells you what is around you. Those two questions are essential for an organism that has to make a decision to approach or to avoid, right? But look where they meet up. They converge in that general anterior cingulate region. So the anterior cingulate has access to what you are perceiving about where you are and what you are. Why? Because it is tasked with the decision-making capability for your brain. We also know that this is extraordinarily important because this excellent study, this meta-analysis of voxel-based morphometry studies of 6 major psychiatric disorders appeared in JAMA Psychiatry in 2015. And here you see that the dorsal anterior cingulate and the anterior insula are dysfunctional in this fMRI data in all 6 of these major psychiatric disorders. But that makes sense because one of the major problems all of our psychiatric patients have is in decision making. And it is in forming healthy attachments. And those psychiatric patients are most often suffering the pains, as Paul Mclean told us, of that most painful mammalian condition, separation. And your job as a psychiatrist is to help them find ways to re-achieve attachment, secure attachment. Okay, so that leads to this thought experiment about allostatic loading really coming from insecure attachment, which is based in this neurocircuitry that evolved to help you achieve those 4 attachments. And it's very important for you as a mammal to achieve secure attachment. When you don't have secure attachment, that's when you have separation pain. That is a driver for psychiatric disorder. So that's really an important sort of hypothesis to keep in mind. So when we talk about where your vulnerability and resilience come from, I like this work of de Cloet. And he talks about a 3-hit concept. Here you have in the upper left-hand corner, he depicts for you certain genes that you're familiar with because many of them are researched in psychiatry, like BDNF, like glucocorticoid receptor, the mineralocorticoid receptor. Oh, what did I do?
Professor, I think that you just closed the presentation. You just have to.
Okay, let me go back. I'm going to put you there. Oh, there it is. Can you see it?
Yeah. Yes.
Okay, good. So, and then you have your early environment, and you see that when they are when you have lack of attachment or traumatic experience in childhood trauma, it's going to interact with your risk genes, your polygenic risk factors, And you may develop a propensity through a programmed phenotype, an unhealthy sort of neuropsychiatric phenotype, so that later on, when you have a separation challenge, you are going to suffer from the loss of attachment, and you are going to develop a vulnerability as opposed to a resilience. And we see this. I'm not going to spend too much time. You'll have these slides, that insecure adult attachment associated with an increased risk of metabolic syndrome. Metabolic syndrome is the foundation for stress-related non-communicable diseases, isn't it? It's hypertension, it's the truncal obesity, it's hyperlipidemia, and it's a relatively insensitive insulin receptor that leads to vulnerability to type 2 diabetes. So that kind of polygenic risk factor analysis put together with a traumatic early environment is going to set you up with a phenotype that's going to lead you to metabolic syndrome. And you can check on this through the childhood trauma questionnaire. And what happens? You see reduced gray matter volumes in that medial prefrontal cortex that I was talking about. And so when you have two adverse childhood experiences, you're going to wind up with a smaller anterior cingulate. And look at how terrible. This is the work of Felitti and Anda at the CDC in the United States. It's horrible when you have adverse childhood experiences. It leads to all of these risks, including 4.5 times the risk of major depression, and 12 to 15 times the risk of suicide. Okay? Bad stuff. We don't have time to go over some of these very interesting molecular biology experiments, but non-coding mRNA like MIR218, an overexpression of this non-coding RNA in the medial prefrontal cortex pyramid neurons promotes resilience to social defeat stress, okay? So this is very interesting because social defeat stress has a lot to do with social attachment that we've been talking about. And then childhood trauma, dependent DNA demethylation, hypomethylation is important because it affects glucocorticoid response elements of this particular gene, and that's associated with increased risk of developing stress-related illness. This is from Maunder and Hunter, insecure attachment. You're going to get more disease and you're going to get more healthcare utilization through these intermediaries. Visceral pain and emotional pain are connected. This shows you the anterior cingulate and irritable bowel syndrome. This shows you pain itself, and there's very little that separates emotional pain of separation from physical pain, which is one reason why it is extraordinarily hard to help patients with chronic pain syndromes. And this just shows you that if you are someone with post-traumatic stress disorder, you're building up the muscle of the amygdala and you're weakening the muscle in the medial prefrontal cortex. It becomes hyporesponsive and there's dendritic atrophy. And fortunately, these structural changes are reversible with our therapies if we're lucky. Hippocampus, as you know, stress will reduce volume and cause dendritic atrophy and reduce neurogenesis, while the amygdala is getting stronger. So this is a common problem in many of our psychiatric disorders, not just PTSD. Resilience, when you look at the work of Steve Southwick and Charney and their group, you see that many of the things that produce resilient children are attachment-oriented. Many of the things that produce resilient adults are attachment-oriented. And at our place at the Benson-Henry Institute for Mind-Body Medicine, we studied the work of George Albee, a famous social psychologist. And he focused on psychiatric disorders, but he knew that there was a numerator, your physiology plus your stress profile, And there was a denominator, your coping skills, your self-esteem, and your social support. And you could get a rough feel for the amount of emotional disturbance and mental disorder you were vulnerable to. His mistake was that he didn't include physical disorders, because the same kind of equation or ratio we believe is important not only for psychiatric disorders, but for all non-communicable diseases. So you can think of the numerator as stress or separation threats, as we argued through evolutionary biology, and adverse childhood experiences as being an example of extraordinarily difficult stress. And resilience, with all of these factors: relaxation response, mindfulness, social support, pro-sociality, cognitive skills, positive psychology, spiritual connectedness, exercise, mindful exercise, nutrition, low glycemic diet, sleep hygiene, healthy habits. If you're successful in building that denominator and lowering that stress through incorporating meditation and exercise and good sleep and that kind of thing, you're going to reduce your allostatic load and reduce your vulnerability. That's really the fourth hypothesis of mind-body medicine. We believe this is true. This is our colleague Ahmed Tawakol, who's a cardiologist and a neuroimager. And he did this wonderful study in the Lancet, and he was able to show that with increased stress and amygdala activation, you activate your bone marrow, you create an excess of myelopoiesis, the production of pro-inflammatory monocytes, producing cytokines because of that conserved transcriptional response to adversity we were talking about. And you wind up with pro-inflammatory states, including the very pathogenic state of atherosclerotic inflammation. And he was able to show this using PET-CT, and now he's using PET-MRI to show the same thing. And he's also been able to show that psychosocial stressors, socioeconomic status, fit into this same paradigm. We've shown that there are changes in heat maps in terms of gene expression profiling. So if you teach somebody, these are novices N1 and N2, these are experienced meditators, and these are the genes on the y-axis, and this is these are cross-sectional heat maps. And you see certain genes are hypoactive, green, certain genes are hyperactive, red. And if you teach a group of novices who had the opposite sort of gene expression profile to the experienced meditators over 8 weeks to learn relaxation response breathing, you can start to change their gene expression. Very powerful. And when you look at these genes, the ones that were underactivated in the experienced meditators were reduction in the innate inflammatory response through reduction of NF-kappa B dynamics. So let's quickly go through. We need to do more in learning how glial cells, microglial cells, and astrocytes interact with neurons. That's so important. Our models are so simplistic in terms of synaptic functioning. We published an article back in the late 90s on macrophages and microglial cells and how important they were. So, and then oligodendrocytes, extraordinarily important. We need to know more about how they age and how stress affects them. Because as you know from your clinical work, if you have patients with periventricular white matter disease or deep white matter problems, or lacunar states, vascular dementia states, vascular depression states, if you interrupt the flow in the medial orbitofrontal, medial forebrain bundle through the subcortex by taking apart those myelinated axons carrying neurotransmitters from the upper midbrain to the frontal lobes, you're going to weaken that medial prefrontal cortical area, and all sorts of bad things are going to happen. Frontal network syndromes, decreases in emotion regulation, etc. So we need to know more about keeping oligodendrocytes as healthy as possible for as long as possible. But I wanted to just spend a couple of moments, if I might, about the mitochondria. And the mitochondria track with the brain in terms of normal stress, tolerable stress, and toxic stress. They have the same inverted U-shaped curve. And you can do protective embedding through exercising and so on, or detrimental embedding through psychosocial trauma, and the mitochondria will suffer. The mitochondria actually, they're like the brain of the cell, and they are taking notice of your psychosocial stress while your brain is taking notice. So you have that triad of amygdala, hippocampus, medial prefrontal cortex that is taking notice of your threats. Your mitochondria are doing it in every one of your cells. And this is the work of Picard and McClean in 2018. I'll leave you with this slide. It's very busy, but it's chock full of important information for us as clinicians. So there's this thing called mitohormesis, and I'm just going to, because in the interest of time, just let me make this point. I mentioned hormesis before. It's biphasic sort of dynamics. And so when you're talking about your mitochondria, they have the same biphasic hormesis curve. So if you were to think about this curve, which is the Yerkes-Dodson curve, the one I showed you earlier about a normal stress, tolerable stress, toxic stress, your mitochondria are doing the same calculus. This would be normal stress, tolerable stress, and your mitochondria becomes more efficient under those conditions. But if you're under toxic stress conditions, your mitochondria becomes less efficient. Actually, they break apart in mitochondrial fission. And that's going to lead to disease vulnerability. It's reflected in the disease vulnerability and decreased performance we see in the Yerkes-Dodson curve. This is from McEwen's group, the late Bruce McEwen. Unfortunately, he died in 2019, our greatest stress researcher. And he's able to show that neurons have the same bell-shaped curve. So as you increase levels of stress into toxic stress, your synapses suffer. They're suppressed. Your adaptive plasticity goes down, and there's loss of neuronal resilience. This is a slide that shows you that in your anterior cingulate, you have the same bell-shaped curve. And when you get into toxic stress, your anterior cingulate starts to perform less well and starts to make bad decisions, what we see in our psychiatric patients. So this is fascinating. And what we're going to try to do at the Institute is connect up these examples of what we believe is the same process being managed at different systems levels in the organism. So really, it's separation threats over attachment solutions. That's what really is leading to vulnerabilities. This becomes important. We have to do more to promote health through mind-body, because we know that we can improve the capacity of that medial prefrontal cortex to dampen toxic stress. And important for modern medicine, we have to spend more resources here doing health promotion through primary prevention, but also secondary prevention and tertiary prevention. This is acute care medicine, which is reactive. This is proactive public health medicine. And the secret of quality is not, the electronic medical record. That's a small part of it. It's really keeping our focus on the fact that the doctor-patient relationship is built on love. It's built on attachment. We're there to try to help patients return to their secure attachment. This slide says, I did it, I found a substitute for quality. That's never going to happen. Quality really is, because we're mammals, we rely on secure base attachment. Quality in the patient-doctor relationship is always going to be about the human connection. Now, just very quickly, look at artwork. The artist is processing what we've talked about, the evolutionary brain process we've talked about. And this is the famous Creation of Adam by Michelangelo. There's God in his brain. Michelangelo's playing a little joke on us. And there's Adam, there's the human being. And Michelangelo focuses our attention on this, the gap, the separation, but he also focuses attention on this attachment. between God and the human being. But he's setting up the separation attachment dialectic, which is our state as a creature. This is Creation of Animals by Tintoretto. I put that there because consciousness is not a categorical factor. It's a continuous variable. So animals have consciousness as well. Even E. coli, if you want to stretch it, has primordial consciousness because it's a sensory motor analyzer effector. This is about a grandparent and a grandchild, and I like this because we have grandchildren, and there is the connection, overcoming the separation challenge. This is from A. Milne, The House at Pooh Corner, and we read this to our children because sleep is a separation challenge for little kids. And it says, so they went off together, but wherever they go, whatever happens to them on the way, in that enchanted place on the top of a forest, a little boy and his bear will always be playing. There's the attachment. And you're giving them a teddy bear as a transitional object, to use object relations theory. And that child feels the securely attached through this transitional object given to them by a loving parent. And that helps them overcome the separation challenge of sleep. So yes, neuroscience. I love this slide of all of these guys trying to understand the human brain, but always in the service of attachment solutions. So this is a famous painting by Sir Luke Fildes from the UK, and there's the father and the mother, and they're worried they're going to be separated forever from their beloved child. And this is the doctor trying really hard to figure out an attachment solution for these loving parents and their child. That's really what medicine has been, and that's really what we hope medicine will always be, because of this great panoramic history of evolutionary brain biology we've shared today. So I'll leave it there. I'm sorry it went over a little bit.
Thank you, Professor. It was a pleasure hearing your lecture. And we have people here on YouTube saying that they were learning a lot. And I think that Professor Euripides wants to start with the questions.
Okay.
Hello, Greg. It's listen to you for me is like an attachment solution.
I'm glad to hear that, Yuri.
If Nicholas Tinbergen was one of your heroes, I can say that you are one of my heroes.
Thank you so much.
And if I could, I would like to present to everybody here. Let's see if I can find. I want to show to all the present, other people that is seeing this presentation, one of Greg's most relevant works. Let me see if I can show. So 5 years ago, we were able to invite Greg to come to Brazil, and he did a wonderful job giving us a course about some of the ideas he presented here today. I think they were updated, but if you want to get into the knowledge or the idea, the mechanisms, the development, the biology, and evolution, like Nicholas Tinbergen suggests, you have to read this book, Compassion and Healing in Medicine and Society. I read this book, and reading this book, I was so impressed how one person could put together knowledge so from so different areas that they interface psychiatry. So the boundaries between psychiatry and neurology, the boundaries between psychiatry and medicine, and sociology, anthropology, and biology, psychotherapy, and all other biological interventions in psychiatry. Greg was able to do that, focusing on this idea, on the nature and use of attachment solutions to separation and challenges. And I have to say that during this pandemic, I always come back to this idea when I was telling to my patients or telling to people during interviews about our work here at our university, about one of the most important mechanisms to help resilience during this difficult period, focusing on coming back to your attachment solutions, looking to this, all the anxiety and threat and fear associated to the separation involved in the pandemic, the virus, and looking for solutions in your most significant relationships. So I did that with my mother, with my friends, with my colleagues, and today a little bit with seeing Greg Stout. So thanks a lot, Greg. I appreciate it. And we did a course with Greg. So Greg, nobody is listening, but if you want to take a screen, take a. So this is the course. Here we have a 5-day course that when we discuss very deeply each chapter of this book, having some Brazilians researchers making comments on it. So. As I talk to you guys, I am committing myself to read this book again and see this course again, because it's really something very nice. Thanks a lot, Greg.
Let me just say that to your younger members of your department, that Yuri here at Mass General, it's well known that he's a phenomenal physician. He's a physician first. And his persona as a physician. People here, I remember when my mentors were still alive, that when Yuri's name came up, they would glow, they would smile because of their experience of him as this energetic, young, caring physician. So I'm so glad that you have exposure to someone like him. And so my ideas about attachment fall on fertile ground because he already knew that. He was intuiting that in his role as a physician. And that's why I think all of us get into this role as being a physician, because maybe we had wonderful nurturance when we were younger and we wanted to pass that on in our role as physicians. But really, my approach is that that's really what's generating the love we have for our patients and why we do what we do. And of course, the pandemic brings that up. I just want to make one other point. In the interim, between the course I did in Sao Paulo, which I always feel was a great privilege, my thoughts about separation have become more balanced. And I now think about separation as obviously it's inseparable from attachment. It's the core dialectic. But I also think of it as a personal trainer, that it's, when we want to get in shape, we go to the gym and we have a personal trainer who gets us through. Separation really teaches us the importance of attachment solutions. And so we need it. And I think that that's why we see that bell-shaped curve, because when we have normal stress and then we have a bump in our separation stress, that can actually help us become more attached. and and that's really where resilience resides. So I just want to put in a plug for separation. It's not evil. When I talk about a separation challenge, that's part of the beauty of life, that we experience separation challenges, and it calls upon our building our capacity to find enhanced and refined ways to be attached. And I think that's the lesson that we all learn when we study individual development, when we read Piaget and Melanie Klein, all of these people talking about individual development, separation and individuation. We have to experience that if we're going to be attached at a higher level, an enhanced and refined level. And so I think that's part, that may be the core part of the mystery of life. And so it's a privilege being a doctor, because you're always immersed in this mystery. So I'll just leave it there. But thank you very much, Yuri, and thank you to everyone. It's been as usual. It's very uplifting for me to be with you all down in Brazil. So
thank you, Greg. Thank you.
All right. So, Yuri, I'll try to figure out how to put the slides in the Dropbox, but I may be back to you, Marina, since you're a young person who knows technology better than Yuri.
I'll try to do that. It's okay. And Professor Yuri have shown us his capacity for, loving and caring for us during the pandemic. So we, the residents, and knows that he's really good at it as a physician and as a boss as well.
It doesn't surprise me. So you're very fortunate. So,
Professor, we have a question on YouTube.
Sure.
Could I read it?
Yes.
So it's late. It's this. I will try to translate it. So it's a physician that works in one of our public mental health centers, and she's asking this. So the more challenging separation events the patient faced during his or her life, for example, homelessness, drug users, not meeting their parents, or struggling to have some food at home, and etc. How does mind-body medicine and how long does mind-body medicine practices will take to build resilience and to bring results as attachment solutions for this group? Like, if there is something that we can do about it, and how long does it?
Yes. Well, I may not have gotten the whole question, but I do. That's a very important question. So I thank the person for asking it. So what we're dealing with when we're dealing with these almost intractable problems in some of our patients, if you think back to that pyramid slide of vulnerability and resilience, there are going to be people who have the 3 hits, and unfortunately, if they've inherited a vulnerability to substance abuse, and then are brought up in an environment without nurturance and with trauma, they are going to find their way because of that anterior cingulate loop. Remember, the anterior cingulate connects up with the ventral striatum, the nucleus accumbens, and ventral pallidum and the ventral tegmentum. Dopamine is a huge part of that basal ganglia thalamocortical loop. That's what I was talking about, valence and the salience network. And so if you're born with those 2 hits, you're going to find substances that reduce the mammalian pain of separation. Okay? And you are going to, and those substances will hijack those receptors, mainly the dopamine receptors and the mu receptors, the mu opiate receptors, in that circuit. Because remember, that circuit does so much to make us human. So not only is the anterior cingulate mediating salience and balancing our experience, it's your brain's reward circuitry. It is also, it is given the responsibility for helping you make decisions, right? We went over that. It has pain, memory, it has salience, and it's your reward circuitry. So if you're in so much separation pain for the reasons I mentioned, you're going to find heroin. You're going to find cocaine. You're going to find some substance that reduces that pain. Because as Paul Mclean said, it's the most painful mammalian condition, separation. And Yank Pangsep did wonderful work looking at that circuitry and saying, when you have social defeat stress and separation stress in a mammalian animal, that maps onto the suffering in the human. The same circuits are involved. And once you hijack a receptor with an artificial substance, then normal attachment pleasure, it goes down. And that becomes the problem for these patients. They have artificially excited those receptors. So the idea of being with their children, we've all had patients, right? Severe heroin addicted mothers. They don't respond to their children's separation cry. They respond to the drug. In fact, my mentor Paul Mclean did these studies with he did it with hamsters and he did it with squirrel monkeys. Very instructive. What he did was he knew the anterior cingulate was responsible for the mammalian behavioral triad of attachment. So he first ablated the anterior cingulate in the hamster mother. She no longer responded to the cries of the hamster pup. She then ablated it in the pup. The pup no longer emitted the separation cry when it was separated from the mother. Same thing with the squirrel monkeys. So then he did another experiment. He doused the anterior cingulate in the squirrel monkey mother. She no longer responded to the separation cry emitted by the squirrel monkey baby. He doused the anterior cingulate in the squirrel monkey baby, and the baby no longer emitted the separation cry. So this is what's happening in our patients. They are disrupting this very natural evolved circuitry that was meant to support our attachments, and they do that because they start life with vulnerabilities, and their environment in terms of upbringing often for some reason has disturbed that structure, leading to disordered function. And so it becomes really hard for us. But hope is there, because we know that the brain is plastic, and if you can find in psychotherapy we call it corrective emotional experiences, right? With a nurturing therapist, cognitive behavioral therapy, we can try to reset that structure. But also, just to bring this up because I didn't stress it in my talk, spiritual connectedness. So it shouldn't surprise us that one of the most successful ways to stay away from drugs after you polluted yourself with artificial drugs is to do 12-step programs. Because really the active ingredient in a 12-step program is the sense of being connected to something greater than yourself. You're connected in a spiritual way through the 12-step program, and that is powerful for that prefrontal cortex, because the prefrontal cortex is all about belief. which is why placebo resides in the prefrontal cortex. So when you, as a caring doctor, say to a patient who comes in, the patient is so worried about, unusual feelings, and they think, oh my gosh, my mother had cancer. I have cancer now because I have this twinge. And they come in and their amygdalas are aflame with threat signaling, and they're all jazzed up, they're all anxious, and the doctor examines you and then puts his hand on your shoulder and says, oh, don't worry about this, I've seen this before, this is going to go away, I know the medication, you're gonna do fine. That's a very powerful biological event for that patient, because that prefrontal cortex gets strengthened to tell the amygdala, oh, you see, everything's okay. And that is part of a belief system that we can access through spirituality as well. So that's a very good question.
Marina, are there any other questions from the audience? Otherwise, I would like to make a short question, if there is such thing.
Sure.
This interesting area. First, thank you very much, Professor Fricchione, for coming back to the Institute, although in this virtual model. It's a pleasure for us, and we had a great talk this time again. My question is if we should, at some point, rethink about the notion of communicable and non-communicable diseases, given the possibility of epigenetic imprinting of separation trauma, and that can be passed on to the descendants of. You have some very nice topics about this in your book, which is really endorse Euripides' words about it. It's a very dense book, but very easy to read, and you make lots of insights from our clinical practice.
Thank you.
So back to my questions, if you could just say yes or no, or if we don't have much time, but about this possibility of communicating our traumatic experiences to our descendants.
Yes. No, thank you for that question, Orestes. It's really important to examine that. And my colleague Lee Schwam, who's a neurologist here, had an essay in the Lancet where he takes your point, and he says that what we call non-communicable diseases are communicable, and he really is reflecting our attention on social network theory, and that many of these stress-related illnesses, if you're in a community that is suffering from the same kind of stressors, there can be a contagion, and so it's communicable through social network theory. But you're bringing up intergenerational transfer of toxic stress, which is, it's powerful. I mean, just think of the revolution in our thinking over the last decade, because we were strict Darwinians, right? No inheritance of acquired characteristics. But epigenetics is neo-Lamarckian. It's the idea that there is the possibility of passing on acquired characteristics, and the idea is through germ transfer, through germ cell transfer. There's the classic study of a famine and how it changes your insulin genetics and that kind of thing, and that's passed on intergenerationally through germ cell changes in gene expression in epigenetics, through hypomethylation and hypermethylation of different genes and so on. So that is going to be a force to reckon with in terms of re-examining our psychiatric disorders. That's really, to the young people, I mean, my brand of psychiatry was very primitive in understanding. And of course, I'm not a big fan of DSM. I do think, we spend way too much time educating residents using DSM.
Gravou lá, né?
I would just say that one of the reasons for the talk today is to focus the attention of trainees on making sure, yes, you can learn DSM because it's helpful. You can put a drug with certain cluster of symptoms and so on, but don't believe that that's scientific. It's useful, but it's not really scientific. Try your best to return to first principles and try always to understand your patient using some of the things we discussed today, especially when the patient's complex. That's the only way you're going to get to some understanding of them. DSM is going to fail you. You can use it to bill. You know, that's what we do in the United States. We use it to bill. But whenever I give a DSM diagnosis, I know it's a fantasy. Except when we're dealing with, you know, a simple major depressive disorder. But today, our patients are so complicated. They have so many different things going on But anyway, I got off on a tangent. The point is that yes, we are going to learn more and more about intergenerational transfer of trauma, of toxic stress, and it's going to be really important for us. The other thing is, we now know more about a placental health. And, you know, we used to take the placenta and throw it away. That was a big mistake, because so much of what gets transferred to the fetus in terms of fetal brain development is part of how that mother is metabolizing toxic stress in her life. Because there's oxidative stress. The placenta is swimming in activated macrophages that are pouring out pro-inflammatory cytokines that at certain vulnerable periods of brain development in the fetus are certainly having effects in the structure of that fetus's brain and how it progresses into childhood and adulthood. So we are going to have to go way, you know, it's not even child psychiatry, it's fetal psychiatry. And immunology is going to be a big part of it. And so, yes, intergenerational transfer, but also maternal fetal transfer that's going to play itself out in the future. But anyway, that I won't be around to see the blossoming of psychiatry, but it's going to be extraordinary. That's my prediction.
Thank you.
All right.
Please.
Anyway, it's great to see these guys.
Thank you so much.
You're welcome. All right, so we'll keep in contact, okay? And be safe. Be safe down there.
Thanks a lot.
Thank you, Professor. It was amazing. Thank you so much.
Bye-bye.
We'll see you. Bye-bye.
Bye-bye. Marina, você tem que missão agora, Marina. Você tem que cumprir essa aula dele pra gente, hein? Tá bom?
Tá bom, pode deixar. E a todos que estão no YouTube, na semana que vem a gente vai falar sobre o histórico do conceito de esquizofrenia com o Rafael Massuda, tá bom?
Bacana. Obrigado pela participação, né? Então tá joia. E não deixem de, quem quiser o link desse curso, fala com a Marina, ela manda o link para vocês. Acho que é um curso que vale a pena assistir. Fundamental, né? E porque, como falei antes, pouca gente, não sei se o Orestes concorda comigo, poucas vezes na minha vida eu vi alguém integrar conhecimento com o grau de profundidade que ele faz, né? É impressionante, né?
Com certeza. E o livro dele, é um livro muito agradável de ler. Apesar de ser muito denso, é tão incrível a maneira como ele conecta os conceitos e você está o tempo todo pensando em coisas que você vê na prática clínica. Então, realmente é uma leitura imperdível mesmo.
Ele falou de alguns caras que influenciaram ele, né? Quando eu fui fazer o meu pós-doutorado lá no Massachusetts General Hospital, né? Eu trabalhei com alguns deles, né? Um que já tinha morrido, que fez o livro Massachusetts General Hospital Consultation Psychiatry, era o Thomas Hackett, que era um cara que dizem incrível, mas eu cheguei lá e já tinha morrido. Mas aí tinha o Ned Kessen que era o chefe do departamento, e o George Murray. Eram psiquiatras, eram padres jesuítas que dedicaram a vida a estudar psiquiatria. E eles discutiam os casos de consultoria psiquiátrica no Mass General com um grau de profundidade que eles sabiam tanto a clínica médica, a neurologia, quanto qualquer outro profissional não psiquiatra. Então, ele tem essa herança, né? E daí ele foi fazer o pós-doutorado dele. Ele ficou 2 anos, acho, trabalhando com o Paul Mclean, né? Que é aquele cara, Mclean, né? Que é o cara do cérebro triuno, né? Que desenvolveu, que conceituou o sistema límbico, né? Então, ele realmente é um cara muito experiente. Nesse Mind-Body Benson-Johnson Institute, né, eles procuram fazer a nova psicossomática, vamos dizer assim, né? Eles procuram fazer essa integração mind and body e utilizar todas as técnicas, tudo que a gente sabe de medicina, de estilo de vida, eles falam isso há muito mais tempo, com muita profundidade, né? Então, vale a pena a gente se aproximar e conhecer mais o trabalho do Greg. Obrigado, Marina, por tocar.
Foi incrível. Obrigada a todos pela presença e nos vemos na próxima semana. Obrigada.
Tchauzão.
Tchau.